Welcome to AP Biology. I'm Gail Fizer from Darnell Cookman Middle High School and Florida State College in Jacksonville, Florida. Lesson overview for today. We'll be talking about topics 5.3, which is Mendelian genetics, and Unit 5, heredity. And go ahead and grab your paper, pen, and calculator because you're going to need it today. By the end of this lesson, you'll be able to explain how shared, conserved, fundamental processes and features support the concept of common ancestry for all organisms. Explain the inheritance of genes and traits as described by Mendel's laws. Perform chi-square hypothesis testing and use data to evaluate a hypothesis or prediction, including rejecting or failing to reject the null hypothesis. All right, for our warm-up. So this is homework from yesterday with Ms. Kate from Meiosis. If you are just joining us for the first time, go ahead and I'll give you a few moments to jot down your answers. Meiosis occurs in many organisms as part of sexual reproduction. Describe 3 differences. So we're going to describe 3 differences between mitosis and meiosis. I'm going to get my clock running. Okay. All right. I think you've had time to jot down a few ideas. Okay, so there were many ways to approach this. If your description was about the parent cell to the daughter cell, then you possibly said a 2N to 2N in mitosis and 2N to 1N in meiosis. Or perhaps you approached it from genetic results. Daughter cell is genetically identical to the parent cell. Daughter cell is not genetically identical to the parent cell, or number of daughter cells, 2 in mitosis, 4 in meiosis, number of divisions, 1 in mitosis, 2 in meiosis, or you could have talked about the mechanics. Homologs don't pair, homologs pair, no synapsis, synapsis, chromatid split at anaphase, chromatid split at anaphase 2, no crossing over, or maybe you talked about the purpose, repair, growth, asexual reproduction, or meiosis forming gametes. But whatever you did, you had to describe the process in both. If you said something about mitosis, you had to say something about meiosis, okay? If you had just went into a description of prophase and metaphase and anaphase and telophase, that would not have earned any points. Okay, so what you need to know. All right, let me just go ahead and do this part and then I'll tell you the other story. All right, the following 4 features support common ancestry. DNA and RNA are carriers of genetic information. Okay, there's our first one. Ribosomes are found in all forms of life. Major features of the genetic code are shared, by all modern living systems, and core metabolic pathways are conserved across all currently recognized domains. So if you can remember, we had glycolysis in fermentation, and so it didn't matter if you had oxygen or not, that there was fermentation going on. We didn't need the mitochondria to do so. So these shared, conserved fundamental processes and features support the concept of common ancestry for all organisms. Okay, so years ago when I was in a genetics class, I had to take the 1866 paper of Gregor Mendel and rewrite it. Take his paper, it was translated in English, but take his paper and then write a modern day research journal article in proper form. So I thought, what would I ever do with that again? And then look, here we go. I have it out today. So in his paper, and I'm just talking about 3 of the 7 traits here. We had flower color, flower position, stem length. There are dominant traits, which was the violet color, axial flower position, tall stems. White was recessive, terminal was recessive, so was a dwarf stem. But notice that this was his F1 cross. So in his F1 cross, there are 100% of each were dominant. So the parents of this F1 cross, so our parental group, was true breeding, okay? So this would make true breeding organisms. Okay, look, we're going to do this. If I have P's, I'm going to put a little line under that for lowercase. Anyway, so when he crossed a true breeding dominant with a true breeding recessive, then he got his F1 cross. And if you notice, you know what's going to happen, right? That every single offspring will be dominant. Okay, now the next table is referring to his F2 crosses. So now he took the offspring of this F1, okay, which we're now going to breed those, and then that's going to give us our F2. So you know where this is going also. Okay, so what we had was, well, let's look at his numbers first. Okay, so with dominant and with these hybrids breeding, he had a 3-to-1 ratio in each of these 3 traits. He did all 7 traits, but here we had the dominant wrong color, sorry. Here we have a dominant phenotype and the recessive phenotype. So there's that 3-to-1. Now this next table has to do with he only had offspring from the dominant phenotype, okay? He didn't put any recessive into the group. And then he had a 2-to-1 in his ratio. And if you'll look, here's 2 and here's 1, okay? All right, so enough of my old tables. So some quick vocabulary. He used the words characters and heritable features. We now call that a gene. And then different traits, so variant traits are now alleles. So a gene can have different alleles, okay, which means different variations of the trait, like the white flower or the purple flower. But specifically today, we're just going to talk about dominant and recessive, okay? Now, dominant determines the appearance. Recessive is not going to have any noticeable effect if dominant is present. We had talked about a hybrid already, that's a cross of 2 true breeding, and that true breeding produces only offspring with the same as the parent, okay? All right, so you're very familiar with this, I'm sure. We have a homozygous dominant. I'm going to change to D's, okay, because the P's are a little bit more difficult to keep up with. All right, so now we have 2 alleles that are dominant, and then we have a heterozygous, and then we have a homozygous recessive. I know you know that already. Okay, so now let's talk about Mendel's law of segregation. So I want you to imagine that, okay, I'm going to draw us a cell. We're in a cell, and right this moment, we're at, excuse me, we're at metaphase in meiosis 1. So the homologous pairs are lined up together in the metaphysial plate. That's where we are. And we have a dominant and a recessive trait, okay? Now, when we have anaphase, what's going to happen? The D and the big D and the little d are then going to separate, or they're going to do what? They're going to segregate. So now they're separating, and this ends up in a cell, and this ends up in a cell. And then in the future, if there is a, excuse me, I don't have any water sitting here with me, I forgot about it. All right, so here we have if we are fertilized with another one that has a little d, then we're going to have this little d is going to be able to be expressed. And so that's the match right here. Oops, sorry. That's going to match this guy right here, okay? If we are able to get it away from the big D, big D. So that's the law of segregation. They're separated from each other. Okay, I know that you're familiar with a monohybrid cross. We have a hybrid here, we have a dominant and a recessive, dominant, recessive, and now we have our homozygous dominant, our 2 heterozygous, and then our recessive. So there is our 3-to-1. And this is what allows that 3-to-1 ratio to happen, the law of segregation. It allows this moment to happen. Okay. All right, so rules of probability can be applied to analyze passage of single gene traits from parent to offspring. Mendel, in his paper, he actually had this uppercase A, let's just say big A plus 2 big A and little a plus little a represents all the characters of this organism. And so as we talk about the laws of probability, we're going to talk about how do we figure out the probability of having what, besides just having this 3-to-1 number in our heads. So if we have a situation where we're talking, if we have A or B, we're going to use plus, okay? And if we have A and B, we're going to use multiply. And the best way for me to teach you to do probability and heredity math is to do math, okay? So we're going to start off with an easy one. We have 5 children, all sons. If the woman gives birth to a 6th child, what is the probability that the 6th child will be a son? So if we started off with this is the possibility of being a girl, and this is the possibility of being a boy. So then what's the probability of being a boy? It's half of these, right? Okay. So that's just, I know, easy breezy, just getting us started. Okay, so now probability that the genotype CCDD will be produced by parents big C, little c, big D, little d, 2 of those, 2 dihybrid, 2 dihybrids. Okay, so here I took this right here, and now I have a C and a C and a D and a D. So if you look at it like this, what's the probability of getting a little c from this combination? It's half, right? 50%. And what's the probability of getting a little d from this combination? Is a half. And this c from this would be a half. And this d from this combination would be a half. So if you multiply all these together, you have 2 times 4 times 8 times 2, 16. So the probability is 1/16 that you could get this from these 2 parents. Okay, one more. All right, achondroplastic dwarfism is a dominant genetic trait that causes severe malformation of the skeleton. Homozygotes for this condition are spontaneously aborted. Hence, the homozygous condition is lethal. But heterozygotes will develop to be dwarf. So I already put this in to save some time. If you have homozygous dominant, you're dead. Okay. If you have big A, little a, then dwarf. If you have a little a, then you're not going to express any of the trait. Okay. All right. So Matthew has a family history of the condition, although he does not express the trait. So here's Matthew right here. This is what it looks like if you don't have a trait. Jane is an achondroplastic dwarf. Okay, here's Jane. So she's going to look like this because she can't look like that. All right, Matthew and Jane are planning a family of several children and want to know the chances of producing a child with achondroplastic dwarfism. So if 3 children are born to Matthew and Jane, so here's child 1, child 2, child 3, what are the chances that the first 2 children will not express the trait, but the 3rd child will be achondroplastic dwarf? So if we have a, okay, so what was the chance of getting a little a from Matthew? It was 100%, right? What was the chance of getting a little a from Jane? It was 1/2. A little a, so the same combination. And then we'll have to get this big A from Jane. So what's the probability? 1/2. And then we have a little a that we have to get from Matthew. 100. So wait a minute, did I mess up? Here's those two, and then that one. Okay, 2 times 2 is 4, and 4 times 2 is what? Is 8. I think I did that right. Let's see. Did I do that right? Yes, I did. Okay, good. All right, so I got kind of confused on how many A's. I have A's everywhere. All right, so you would keep this 1 because it's totally 100% if you had a question about that. So if we wanted these 3 kids, that's when they have to be multiplied. All right. Okay, so now Mendel's law of independent assortment can be applied to genes that are on different chromosomes. So now okay, I want you to imagine again, we're in a cell, and we are lined up in the middle at the metaphase 1 of meiosis 1, and we're at the metaphasial plate, and we're going to then pull to the side and split into 2 cells. So this would end up in a cell together, and this would end up in a cell together. So this dominant and this dominant would be together, and this recessive and this recessive would be together. Would this have to be like this? No, this does not have to be like this. This dominant R could be over here. Okay, if this is a different cell that we have and a different option. Okay, so now this is totally random. This is the independent assortment. These chromosomes can assort independently of each other. They do not have to stay together. So now I have the dominant trait of this one with the recessive trait of the other gene. So now this would be in a new cell, and then this would be in a new cell at the end of meiosis. So they would be totally different options, independently assorted. Okay, so let's do a dihybrid cross, and then I'll tie it together with independent assortment. So if we have two traits and 2 hybrids, okay, for each trait. So let's find out what our possible genotypes are going to be. So we can have a big D, big A, right? And we can have a big D, little a. We can have a little d, big A, and we can have a little d, little a. And since those were the same, okay, we can just repeat that. Okay, so now let's just check. So this was, so I make sure that you know how this works. So here's a D, big D, here's a big D, there's big D, little a, little a, little a, little a. Okay, so I already did this so we could, for the sake of time. And now let's count. There's 1, 2, 3, 4, 5, 6, 7, 8, 9. There's 9 that have dominant trait. There's 1, 2, 3 that have a dominant and then the recessive trait. And then there's 1, 2, 3 that have a recessive and then dominant trait, and then 1 that has recessive. So this dihybrid cross, if you have a dihybrid cross, you will always have a 9-3-3-1 combination, and it will be because of this law of independent assortment will allow this to happen. Okay, now, and fertilization involves the fusion of 2 haploid gametes, restoring the diploid number of chromosomes and increasing genetic variation in populations by creating new combinations of alleles in the zygote. So this is a meiosis drawing that I did for my class, and I use it every year. But let's start off talking about this right here. Oops, I got to move something. I was practicing earlier. Okay. All right, so if we start off with this diploid, we have 3 homologous pairs of chromosomes. We got 1 from mom, 1 from dad for each of these. Now, and we're going to go through meiosis. So when we have interphase, here's the 1 from mom, here's the 1 from dad, and replication occurs. Still 3 pairs of chromosomes. Now, if you'll look closely, now there's some variation that has happened on each one. So that was from crossing over. So we can say some variation happened here. And then they're going to align in the middle. And then we have, does this yellow one have to be there? No, it does not. It could be on this side, or this red could be on this side and the orange over there. So independent assortment happens. So there's more variation. So 2 places genetic variation occurs. Anaphase happens, 2 cells, and here we are at our next 2 cells, about to go through the next phase. So here's our 3 cells, and then they're going to line up in the middle, and then they're going to be separated during anaphase 2, the sister chromatids are, and then we have 4 genetically different. If you look at them, each one of them is totally different than another. So if you had, when fertilization occurs, if you had another very diverse, genetically diverse from their parents, offspring, think about these 2 starting off at this location and then going through this process. So genetic variation just continues to increase and increase through each generation. All right, so I'm going to run over this just a little bit. I think you'll be doing some more of it tomorrow, but so let's just get some groundwork done. So you have sex-linked inheritance, okay? And we'll work with this a little bit in some questions. So let's do hemophilia, okay? All right, and that is sex-linked, and that means that it's going to come from the mom's X, okay? So let's do, there's a female, and let's use, here's a male, and let's use a uppercase H for normal, okay? A normal allele. And then we'll use a little h for the diseased allele. So, or the mutated allele. So let's do this. And then we have all right, so our 2 girls, we have both 2 normal girls, okay? They don't express, this one doesn't express any of this trait because she has a gene that will produce cells and proteins correctly, okay? And so, but she is a carrier, okay? As in this mom was a carrier. Now on the boys, we have a normal allele, and then now we have the mutated allele, okay? And the only proteins that can be made from this gene are the one with the mutation. So there's not any other normal proteins to be made because we don't have that uppercase H here, okay? So that's how it's sex-linked, and that's how it affects males. Does it have to be 100% just males? No, we could spend some time and go through different scenarios and come up with situations where we could end up with a female. Okay, so now let's talk about pedigrees. And I'm going to do pedigrees just like I did math. We're going to learn by doing, okay? So the pattern of inheritance can often be predicted from data, including pedigree, that give the parent genotype, phenotype, and the offspring genotypes, phenotype. So let's read what it says first. In the pedigree above, squares represent males. So this is going to be for male, and circles represent females. Individuals who express a particular trait are represented with shaded. Okay, so this will be, they have the trait. Now, which of the following patterns of inheritance best explains the transmission of the trait? So let's go through some questions that you would ask yourself about this diagram, and then you'll be able to answer it. First, are males and females equally getting this trait? And we see that yes is the answer to that. So we can automatically say this is not sex-linked, okay? And we have total, you either have the trait or you don't. So we're not going to do incomplete dominant. And now we have an autosomal recessive and an autosomal dominant. So next question is, do we see it in each generation? Yes, we do. And do we see it from any carrier parents? Okay, so do we have anything that looks like that? No, we don't, okay? So that makes our conclusion, it's going to be not recessive, it's going to be dominant. And the word autosomal is referring to body chromosomes, like pair. In us, it would be pair 1 through 22. And then we would have our sex chromosomes, which would be pair 23. So that's where this word autosomal means body chromosomes. All right, so let's do another one. So let's take a look in the pedigree above. Circles represent females, squares represent males, and shaded figures represent individuals expressing a specific trait. So consistent with that generally. All right, the expression of this trait is most likely due to which of the following. So we have, all right, so let's look at our boys and girls. Do we have boys and girls getting this? Yes, we do. So let's get rid of those two. All right, do we, let's see, that's either you do or you don't. So we'll get rid of that. All right, and then do we have carrier parents having children with the trait? Yes, we do. There's a situation right here. There's one right there. Here is one right here. So now this is autosomal recessive inheritance. Okay, here is a question about Calico Cats. This is my Calico Cat. Her name's Callie. And a student of mine, I'll give a shout out to Amber, gave me this little precious thing when she was 4 weeks old. All right, a gene that influences coat color in domestic cats is located on the X chromosome. A female cat that is heterozygous for the gene, so this is what a calico looks like, has a calico colored coat. In a genetics experiment, researchers made a calico colored female, mated a calico colored female with an orange colored male. Okay, so this is an orange male to produce an F1 generation. The researchers record observations for the cats in F1 generation and plan to use the data to perform a chi-square goodness of fit test for a model of X-linked inheritance. The data for the chi-square goodness of fit test are expressed in the table. Okay, so let's look at this. He expected an equal number of each. Let's go ahead and determine why he expected that, okay? Because I just don't want to take it for granted. So there's a calico option. Oh, what am I doing? There's a calico option. There's an orange girl. So calico girl, orange girl, black male, orange male. Everybody's different, right? So you have one here, one, equal distribution. So that's where this 10 number in each of these came from. Now, the researchers calculate a chi-square value of 4.6 and choose a significance level of p equals 0.05. Which of the following statements best completes the chi-square goodness of fit test? So they gave us, we didn't have to do the math, although I did the math on the next page so you can see it. All right, so here is our, what do we have? We have 4 categories. So our degree of freedom, remember, is our categories -1, so our category is going to be 3. And then we have a 4.6 is our chi-square value, and it is less than, so see, we have 11.34, 7.81, so 4.6 is less than our critical value right here. And here's our, so it's heading in this direction. And so our p-value, there's p.01, there's a value of 0.05. And so this is going to be greater than 0.05. So now we know about our probability. It's greater than 0.05. So what we could go ahead and write when we know that, p is greater than 0.5. We can say that we are not going to reject our null. We can say this is not statistically significant. And we can say that any variation is due to chance. When I'm writing about a null, or I'm writing about the null and a p-value, I write all of those things to make sure that I have everything I need. So now let's compare it to these answers. The null hypothesis can be rejected. Did we say it could be rejected? No, we said do not reject. The null hypothesis can be rejected Nope. The null hypothesis cannot be rejected. Okay, I'm good with that, but it's there too, because the chi-square value is greater than the critical value. Is it greater? Is it this way? No. So D, the null hypothesis cannot be rejected because the chi-square value is less than the critical value. Okay? All right. And so in case you were interested, I went ahead and did this math because the first time I practiced this lesson, it was 2 hours long. And I know my director person would have had this, she would have had a cow. Anyway, I went ahead and worked out this math. And so here are the expected values. Here's the formula. So expected values I inserted, and then the observed values, and then did the math, came up with the 4.6. So I agree with this, what this question came up with. Okay, and there we go, and D was the answer. Okay, so among others, some of the science skills that we're going over today: perform chi-square hypothesis testing, use data to evaluate a hypothesis or prediction, including rejecting or failing to reject the null hypothesis. Some task verbs we're going to use today: construct, predict, determine, and explain. All right, so I know you've seen this question, but not in this particular context. This question has like 4 different parts to it. And so between Ms. Kate and I, we're getting our bang out of this buck. Okay, so in certain species of plant, the diploid number of chromosomes is 4. So 2n, so it said diploid 4, 2n equals 4. Flower color is controlled by a single gene in which the green is dominant and the purple allele is recessive, okay? Plant height is controlled by a different gene in which a dwarf allele, D, is dominant, and a tall allele is little d. All right, individuals of the parental P generation with genotypes, here we have a true breeding, okay, and true breeding for 2 traits, were crossed to produce an F1. So what's going to happen if we cross those? We already know that if we cross those, that we're going to have this, okay? We're going to have a, excuse me, a big G has to be given from this parent, and a little g has to be given from that parent. A big D has to be given from that parent, and a little d has to be given from that parent. So our F1s will look like this. Now predict the possible phenotypes and the ratios in the offspring of a test cross between an F1 individual and a recessive individual. I did this part of this question because I wanted to go over test cross with you. Okay, so if we have, these are our parents, we're going to have this as one parent, and then we're going to test cross it with this. So I'm going to build a little box, not 16 squares this time because I don't need them. So what's the only option for offspring I have here? It's going to be a little g, little d, right? And so here I have big G, big D, big G, little d, little g, big D, and little g, little d. Okay, so when they are, when they're crossed, we have this. I think I did that right. Okay, so we end up with a green and tall, green and tall, green and not tall, and purple and tall, and purple and not tall. Okay, now we have one of each. So we have a 1 to 1 to 1 to 1 ratio. So when you have a dihybrid, and you're test crossing it, this is always going to be your ratio when you have a test cross from this with this with this. Okay. All right, blank page because I thought I might need it. So here's our scoring guideline. We had 1 green dwarf, 1 green tall, 1 purple dwarf, and 1 purple tall. All right, so we're going to go through. I did a lot of the parts in advance on this because it's quite lengthy. So In fruit flies, the phenotype for eye color is determined by a certain locus. E indicates the dominant allele, and little e indicates the recessive allele. The cross between a male wild type fruit fly, and wild type means normal, and a female wide-eyed fruit fly produced the following offspring. So we ended up with 45 wild type females and 55 wide-eyed males, and some odd thing happened there, okay? The wild type and the wide-eyed individuals from the F1 generation were then crossed to produce the following offspring. So we started off with a parental group that we don't know, okay? And then we have our F1, and now we have F2. So the first thing that it wants us to do is determine the genotypes of the original parents, okay? So to do this, this is what I had to do. I had to kind of work this backwards. So if I have let's go ahead and put males and females, and I'm following this up here, okay? Okay, I got this. All right, so now this was a wild type male. So let's use a big E because it's easier for us to keep up with. And we have a wild type female, but we don't really know something, do we? We don't know if she is this or if she is that, right? A wide-eyed male, so we know he has a little e, and then a wide-eyed female, which she would have to have both. So if we worked this backwards, then we could put our boys in. And let's grab our other boy. All right, so if we know we have a Y here, what would have to be here? This would have to be here. This would have to be here, okay? Now let's put our girls in. If this is here, okay, then this would have to be here. Okay, so these are those offspring, so their parents, these right here. Okay, so now we're moving these. Wild type female has to be this, and this wide-eyed male has to be this, okay? Now, we need to know their parents. What had to happen to make that happen? We could work it backwards. And let's put in, both boys look the same, right? There's only half and half. So here's our girl. All right, so if we have a Y here, we have to have a what? We have to have an X little e, and to have an X little e there, to have an X little e here, then we would have, and here, we'd have to have this. So this is our parents. And there may have been 3 different easier ways to do what I just did. But I want you to see where it's coming from. Oops. Now, the rest of the question will be, you'll be glad that I did all that. And explain your reasoning. You may use Punnett squares to enhance your description, but the results from the Punnett squares must be discussed in your answer. So this alone would not suffice as an answer, okay? You would have to go through all those words that I just said to get to this point. Those would have to be in your explanation of how this happened. Now, what's our next part? Use a chi-squared test on the F2 generation data to analyze your prediction of the parental genotypes. So we're going to use F2 information, okay? And all these are different. All these different? Yes, they are. Okay, and then we'll show all our work. So I'm going to flip to the next page, and that's where we're going to do our chi-square. I have all of this there also. Okay, so now remember we had in the Punnett square, we had an equal probability of getting each. So if we had 100 of the flies, then we can expect 25 of each. So here we have 25, 25 in all of our expected locations, right? And did the math, and it ends up to be our chi-square value is 2. Now, what else would we say now that we know our chi-square value is 2? We have how many categories? 1, 2, 3, 4 categories. So our degree of freedom is 3. We have a chi-square value that is less than our 7.81 critical value. Okay, so it's heading that way. Our p-value is greater than 0.05. So we can say the same thing again. Our p is greater than 0.05. We are we're not going to reject the null. This is not statistically significant. This is any variation is due to chance. Okay. I set in on a thesis defense today of a student that was mine, and I heard her saying most of all these words that have stuck along, been included in college and master's program. All right, so now we have, did we say all these things? Did we come up with the boy being X, big E, Y, and that being the girl? Is that what we said? Let's look. Yes, okay, you're right. Now, did you discuss? I told you to discuss, like all those words I was saying out loud, those would need to be written out. You can support it with your Punnett square, but you can't just have a Punnett square in that situation. Now, did we talk about sex-linked, X-linked genes? No, we didn't talk about that. And we did it, but we didn't say the words out loud. And we talked about what was dominant. And then in the explain portion, we came up with a 1-to-1-to-1. We did the math, which was a 2. And then we talked about all the right things to say. Do not reject the null. You could also say the null hypothesis is supported. It's not my go-to language. This is. And then what else? Anything else we could have said? All right, and due to chance. Okay. All right. Now, debrief and summary. Today, takeaway for today. I hope you feel comfortable with what the law of segregation, independent assortment, and probability, what those mean, okay, and how to use probability and how to use the math of probability. I hope you feel comfortable with reading a pedigree, asking yourself those elimination questions to try to figure out what you're looking at. You know, does it affect boys and girls? Is it in each generation? Do we have some parents that are carriers? And if it's sex-linked, is only boys being affected? You should also be able to explain how shared, conserved fundamental processes and features support the concept of common ancestry for all organisms. Explain the inheritance of genes and traits as described by Mendel's laws. Perform chi-square hypothesis testing. Use data to evaluate a hypothesis or prediction, including rejecting or failing to reject the null hypothesis. And this right here, and it happened, I've seen it happen in my classroom, is why I grabbed this. This is not the end all be all of all ratios, okay? It's not the answer to everything. So if you're talking about a test cross, remember with our scenario, then it is with, there's our test cross. And why would we need to do a test cross? To get to what kind of, which organisms are homozygous dominant and which are heterozygous dominant, because they both have the dominant trait. Now, in probability, remember if it says or, you're going to add them. I couldn't find a good example, and I'm a horrible test question writer. You wouldn't want that to happen. And so here's and so we're going to multiply them. All right, so for our homework tonight, this will be start up as your warm up on tomorrow night's lesson on non-Mendelian inheritance. Blue flowered African violet of unknown ancestry, self-pollinated and produced 50 seeds. The seeds germinate and grow into flowering plants. Of these plants, 36 produce blue flowers and 14 produce pink flowers. What is the best explanation for the pink flowered offspring? Now, you have some, I think this is an easy breezy question. A, blue flowers are incompletely dominant to pink. B, pink flower color is recessive to blue flower color. C, pink flower color is a result of somatic mutations in the flower color gene. And D, a previous generation of the blue flowered parent I think this should be must have included 50% pink flowered plants. Now, use these numbers, okay, to figure out the answer to this question. All right, and then we have a few more nights of saying this. You gotta do this by the 24th, that's tomorrow. Device and internet access. If you don't have a device, if you don't have internet, please reach out to cb.org/tech, your to-do list. And I'm gonna add as we go right here, okay? So device and internet, okay? Check. You should do this. Make sure your email is correct in your College Board account. And you might have put it there when you're in the 9th grade, and you might not be in the 9th grade right now. All right, and tell your teacher you're testing. I've asked all of mine. I haven't been able to get a hold of a couple of them. Check your phone number also. That was probably put in when you were very young also. So check that. Now, if your test ticket is going to be emailed to you, but it's also going to be in your AP Classroom account, okay? So everybody can breathe again about that. It'll be there. If it's not, it'll be both places, okay? All right. So thanks for watching. I hope you'll join us again. Tomorrow will be Miss Kate, and she'll be reviewing non-Mendelian inheritance patterns. And that's it. Not 2 hours. What do we do? 45 minutes. Bye.